WIN was founded in 2010 as a dream of trying to bring together people from academia and the pharmaceutical industry and technology and non-for-profits, all of whom had an interest in speeding up the movement of science into the clinic in the area of personalized precision medicine. And it was, I think, a very good idea. It was hard to get started. I think we are pretty close to being firmly established. We now have 39 members, including representatives of all of those stakeholders that I mentioned. And these symposia are terrific. It's very unusual, I think, to have speakers from industry and speakers from academia alternating randomly on the platform, and all speaking about research in its fundamentals and exchanging ideas and asking questions of each other. And I find that very stimulating and something that I think will move the field forward. And we have launched our first clinical trial. It's very innovative, and we have more planned. So I think the scorecard is out, but I'm an optimist. The WINTHER trial has been going on for about a year. It was a little slow to get rolling because of the complex logistics of the work that has to be done on the human tissue in order to select therapies. But I think it's rolling pretty smoothly now. We have one more hurdle. Two of the participating institutions in the United States need to wait for FDA approval. That wasn't required in Europe or in Canada. Well, I think over 100 patients have been screened, and some dozens of patients have actually participated so far. The target is 200, so we need to grow a little faster, and that's in the plans. Today, so far in the morning session, we had a terrific discussion of immune factors that affect the response to cancer therapy. Not only immunology directly killing the tumor, but immunology interacting with chemotherapy and with standard targeted therapies. And enough has been learned now about how to assess the immune response in an individual patient, so that I think in the next year we're going to see huge amounts of information coming out on each patient. Not only detailed description of the genomic aberrations in their tumor, but detailed descriptions of the immune system in that patient and its capacity to respond to the tumor and what might be blocking that response. We've learned that targeted agents can produce strong responses, but cure is the goal. And cure has been obtained with surgery and radiation and with chemotherapy of the standard variety. In the case of chemo, it usually takes combinations, and we feel it is very likely it'll take combinations of these targeted agents to actually achieve cures. The challenge is there's a couple of hundred potential targets. There's nearly 1,000 potential agents, and which to combine with which is should be worked out rationally and intellectually rather than just randomly putting things together, because we'll never achieve anything. So the work now involved in developing combinations is trying to create algorithms and analytic tools that help us pick the very best of 2 or 3 drugs to combine together. And then we always have to worry about combined toxicities. So it's still hard work moving from the concept, let's combine therapies, to moving to an actual clinical trial. Well, this afternoon I've been invited to provide sort of an overview, an update of our experience at MD Anderson. And beginning in March of 2012, we set out to sequence 3,000 tumors in patients where standard therapy has failed and where they are candidates now for experimental therapies to try to pick the appropriate therapy that matches the genomic abnormality in their tumor. And we have achieved that goal. We've sequenced over 3,000 patients now, which just the infrastructure to put that together was a tremendous amount of work, and to the credit of the institution. We're learning a lot from this. We're finding about 35% of the patients have potentially actionable aberrations in genes in their tumors, but we're also finding it's very hard to match them to a drug against one of these genetic aberrations, either because the drug's just not available at all, there are no drugs, or the drug is involved in certain clinical trials and is not available for that gene abnormality in other patients, or in some cases the drug has been approved, but in only certain types of cancer where it's been shown with clinical trials that giving that targeted agent produces responses in that particular kind of cancer. And this is very frustrating because patients know they get on the internet and come in pretty up to date on what's going on. And when they find out they have a particular genetic aberration in their tumor, because we share the data with our patients, they say, well, let's get a hold of this drug or that drug that's been approved. And the sad thing is it's only been approved for breast cancer, and they have colon cancer. And these drugs are very expensive. So we have to work out mechanisms to investigate patients like that and pool the data and find out whether the drug works, studying 20 or 30 patients, so that in the future, they'll have access to these drugs paid for by insurance the way it should be. Oh, it's going to take another 5 years probably of hard work, maybe a decade. I think in 2015 we're going to see, there's been a stimulus, especially in this meeting, we're going to see more and more trials that involve innovative combinations. Not only innovation in the drugs that are new, experimental drugs, but innovation in the way the drugs are selected. And I think this is very exciting. We're putting together the best minds in the pharmaceutical industry and in some of the top academic medical centers that are members of WIN, so that we can try to make this happen. And then we need to identify more biomarkers, and there are going to be more trials that take tissues and especially take blood samples and try to determine biomarkers in the blood that can detect the presence of cancer, detect its disappearance, and detect its recurrence, and hopefully detect whether that patient might respond to certain drugs. And that is, I think, that's not unique to WIN. Everybody realizes that sampling the tumor can't be done at the level in which we need data, and we must learn how to sample the blood and get that kind of information from pieces of DNA or pieces of the tumor that have spilled over into the blood.